All right, this is part 3 of the review for exam 2, and at this point I'm moving into the section covering specific pathogens. My plan for this section is I don't really see a big benefit in me taking the time to go through every single individual bacterium on the list. A lot of them are sort of straightforward information that's listed on the slides, and me reading off the slides just doesn't make much sense. So what I'm going to do is give a quick introduction, and then I'm going to take the time to go through the handful of bacteria that I have a lot of detail on, the ones where we go into the specific virulence factors and their mechanisms, and then the other ones, obviously I'll go through in class, and I think you guys should be able to study on your own. So the first thing to point out, and I mentioned this in an earlier section in the notes, is you have to be careful when distinguishing the naming system here. The name of the bacterium and the name of the disease don't always match. Sometimes they match really closely, but they're not identical. And so make sure you're careful about naming the pathogen itself and naming the symptoms. Okay. There are lots of examples of this. So for example, somebody, it's commonly said that someone has a tetanus infection. The name of the bacterium is actually Clostridium tetani. Clostridium is the genus, tetani is the species. Tetanus is the name that we give the symptoms. So close, but not identical. The other thing to be careful of is that just because a particular bacterium has a name that sounds like disease doesn't mean that it's always and only the cause of that disease. The most common example of this is pneumonia. There are species of bacteria that have the species name pneumoniae, which is obviously very similar to pneumonia. There are lots of different bacteria that can cause pneumonia, and even viruses. And so just because something has the name of a disease doesn't mean that it's the only cause. It just means that it is one of the many pathogens that can cause that type of infection. The other thing to remember is just because a bacterial species has a scary name, like pneumoniae or tetani or whatever it might be, that doesn't mean that that bacterium will always cause those symptoms in that person. Most bacteria are, to some degree, opportunistic. Some are more likely to cause infection than others, but there are lots of people that have, for example, strep throat. Everyone knows, the symptoms and how terrible it is, how uncomfortable it is. But there are plenty of people, millions and millions of people that have strep, the same strep that can cause the actual infection growing in the respiratory tract, but not actually causing symptoms. And so things aren't quite as cut and dry as you would hope they would be. All right, when you're studying this section of the notes, the way that it's best to do it is pick out the most important information, or what you think is the most important information based on what I say in class and your interpretation of the notes. And these are, in general, the topics discussed. For every single pathogen, we should, you should definitely have this. Gram status and shape will be there. The rest of these, most of this information is there for most of the pathogens in the notes. If not, I'll probably mention it when we go through it during class, but don't stress too hard over it. For the bacteria where we don't go into a lot of details, I'll start off with Staph epidermidis as the first example of that, and then I'll move on to the more detailed ones. So for bacteria like this, where I don't go into a ton of detail, I'm just giving you, sort of a basic rundown of the species, where it lives, what it's doing. When you're studying these for the test, what you should expect to see is these bacteria will be tested on multiple choice questions, and the thing that I'll most likely do is I'll either give you the species name in the question. So remember, Staphylococcus is the genus, epidermidis is the species. And so I may say, which of the following best describes Staphylococcus or S. epidermidis? And the correct answer would say something about, a salt-tolerant bacterium that's a normal part of most everyone's skin flora that lives on the surface of your skin. If I switch it around the other way, I might give you that description, a salt-tolerant bacterium that's a normal part of skin flora, and then ask you to pick from a list of different bacteria which one matches it. The point is, for these, I'm not picking out really, really fine details. That's not the point of it. And so you don't have to memorize, necessarily that epidermidis forms clusters. I'm not just going to say which of the following bacteria forms clusters. That would just be too sort of subtle and picky. And so for these, I think making flashcards is effective. Write the species name on one side and 2 or 3 interesting bits of information on the other side, and just be able to pair the two with each other. For the species that I go into a lot of detail about, specifically the ones that have this heading, virulence factors at the top, these I expect you to know more details. And there aren't a lot of them. Only maybe 15 or 20% of the species that we're going through we're getting into details on. But you should be ready to recognize in multiple choice questions the mechanism of how these virulence factors are working. And for the written part of the test, you should be prepared to have a very clear written explanation of at least one of these virulence factors to use as an example of the sort of the general principle. So I'll start going through these one at a time, and I'll start with anthracis. Again, genus and species, Bacillus anthracis is the species. We say that anthrax is the name that we apply to the symptoms when somebody has an anthracis infection. And the way that it exerts its effect is it essentially lyses cells. And lyse just means to rupture or burst apart. This happens in white blood cells most potently, and the mechanism's on the next slide. And what I want you to see is this isn't just some accidental thing. It's not like, anthracis, the bacterium, isn't just some generally bad thing that makes your white blood cells get sick and die. It's a very specific target that makes it happen. And if you think back to virulence factors, what we said is they're pretty much always proteins. And the reason that is because the only thing bacteria really have is their DNA. And the only thing that DNA can really do is code for protein. And so by necessity, the toxins that these bacteria secrete have to be protein-based. In the case of anthrax, it gets a little more complicated because it's actually 3 separate proteins. And so anthrax toxin is really a combination of 3 different proteins, and all 3 are required to actually kill cells. And I'll go through them one at a time. The first one is called protective antigen. And protective antigen binds a receptor on the cell surface. Now, when I say on cell surface, that means host cell, so that's the human cell. And what that does is basically serves as a docking site. So this little guy here, PA, protective antigen, binds to a receptor on the surface of, this would be a white blood cell, right, inside of a person. That's the cell membrane. And that serves as a docking site to recruit these other 2 proteins called edema factor and lethal factor. If you remember from a past class, edema just means swelling, water, fluid accumulation. And in order for this to work, edema factor and lethal factor have to get inside the cell. The whole purpose of protective antigen is to trigger endocytosis. Okay, so the whole bacterium isn't necessarily getting inside the white blood cell, but the toxins do. And so what you get is this complex protective antigen bound to clusters of edema and lethal factors. That complex essentially tricks the cell into endocytosing the whole thing. And from there, these proteins are really just messing with cell signaling inside the white blood cell. Okay? They start binding to and damaging signaling proteins inside the white blood cell, throw off levels of this second messenger. You've probably heard of cyclic AMP. It's just a modified version of ATP, actually. That starts disrupting salt levels, which disrupts water homeostasis. Cells start swelling. In addition to that, lethal factor is essentially an inhibitor that turns off a really, really important family of enzymes inside white blood cells. Kinases, if you've had them from a past class, these are signaling enzymes that phosphorylate other proteins inside of a cell. And turning them off essentially is like turning off the master control circuit inside of a cell. Everything gets shut down, the cell completely stops functioning, and between losing the function of its intracellular signaling and taking on lots of water, the cell just bursts and lyses, and the whole thing breaks apart. And so what I want you to take from this is, it's not like something that's very generically bad, like alcohol or bleach or things that we use to sterilize and kill cells normally. It's very, very specific. Right? Specific proteins from the bacterium disrupting specific proteins, signaling molecules inside the host cell. End result is cells lyse and die, which can lead to really horrible systemic problems, which is why anthrax is such a concern if you're exposed to it. All right, the next one that I spend some time on is Clostridium perfringens, and it's a fairly, not incredibly common, but not unheard of source of food poisoning. And because it's an anaerobe, right, it normally ferments the tissue that it's infecting. One route of treatment can be oxygen exposure because, like I said, it is an obligate anaerobe. The interesting thing that goes with this, and I want to just preface before I say any of this, this is fairly new and really not entirely certain at all, but it's very interesting. There's some evidence, very preliminary evidence, that perfringens, Clostridium perfringens infection, a specific subtype, serotype called type B, as opposed to type A, is possibly connected to the development of MS, of multiple sclerosis. And I'm going to take you through this, but again, the caveat is this is not nailed down, this is not making its way into medical textbooks. It seems like there's some kind of link, and I want to show you the evidence because it's fairly interesting. So what these researchers did is a couple things, and none of it really constitutes a very carefully done experiment, and you can't really do a careful experiment here. This is one of the best examples of why sometimes you just have to go with epidemiology. Right? This would be really easy to figure out if we were just willing to purposefully infect people with Clostridium perfringens type B and see if they get MS. But obviously, no one's going to do that. That would be horrible. And so we're left sort of doing detective work. And what these researchers did is, thing one, they had a patient who was presenting her first symptoms of MS. She was just being diagnosed. And while that was happening, they found that she had Clostridium perfringens type B infection, and they isolated the bacterium that was causing that infection. Right, that by itself is not very strong evidence because it could just be a complete coincidence. So they looked for a little more evidence. The next thing is that perfringens type B, the reason that it's so such an interesting possibility for being involved in MS is the thing that makes it different from other types of perfringens is it produces a toxin called epsilon toxin. This is the potential virulence factor that we're looking at. The interesting thing about it is it seems to be very attracted to and able to cross the blood-brain barrier. Most proteins can't do that. So epsilon toxin binds to and crosses blood-brain barrier and sticks to myelin. Oligodendrocytes are the cells that produce myelin in the CNS, in the brain. Multiple sclerosis is a disease of myelin degeneration. And so the next thing they found is the toxin that the bacterium produces crosses the blood-brain barrier, sticks to myelin, and that makes it interesting. Right? They haven't absolutely proven that the toxin causes myelin degeneration. That would be a very difficult experiment to do, almost impossible in humans. But it's interesting. Right, further, and I guess I should just jump ahead and show you this. This is the evidence for that. You can't do this in a human, but you can do it in a mouse. And so what they did is this green tissue is showing you myelinated neurons, this red signal, they treated, this is mouse neural tissue. This ETX is epsilon toxin. This Alexa 594 is a fluorescent molecule. 594 is just the wavelength of light that it emits. That's a red wavelength. Alexa is actually the name of the guy's daughter that developed the fluorophore. And so link the toxin to this fluorescent molecule, inject it into a mouse, and what you see is everywhere that there's myelin, through here, that toxin binds to and sticks. And when you lay the two images over top of each other, the spots where it's sort of yellowish-orange, that combination shows you the red and green co-localize. Everywhere where you see myelin, you see the toxin. So very provocative idea, very attractive sort of mechanism. Could this toxin be causing myelin degeneration? Okay, the last thing they did is, to try to nail this down, they took MS patients and healthy controls. Okay? And this is after the fact, so they're not exposing them to these things. But if you compare people with MS versus healthy controls, what they found is that parapharyngeans type A was much more likely to be found in healthy controls, whereas epsilon toxin, which indicates type B, was 10 times more likely to be found in people with MS. Right? So it's not an all or nothing thing. But what this suggests is people that were exposed to type A are more likely to be healthy and not have MS symptoms. People that were exposed to epsilon toxin at some point in their life through a type B infection are much more likely to have MS. Okay, so at this point, if this is really a connection, what it tells you is either the epsilon toxin is causing the MS. The other option is, since they're not proving cause and effect, it could be that the MS is causing the infection, but that seems really, really hard to figure out how that would work. It seems unlikely. The last possibility is it's just a fluke, and we don't know why they're connected. But given these data showing binding to myelin, there's a lot of research going into finding out if this really is a real connection or not. Okay, next virulence factor on the list is produced by C. diff, Clostridium difficile, and they're called TCDs for Clostridium, toxin from Clostridium difficile, and it produces two of them, A and B. So TCDA and TCDB, these are the virulence factors produced by C. diff. And the thing that they do is they're enzymes that are, it's kind of a big word, but it makes sense if you pick it apart. The toxins are proteins, and they're enzymes that are called glucosyl transferases. And ACE always tells you that it's an enzyme. Glucosyl means glucose. Transfer makes sense. They're enzymes that transfer glucose from one molecule to another. And what this essentially does is these enzymes get inside of a cell, and they start tacking glucoses onto lots of different proteins inside the host cell, inside the human cell. And that just wrecks everything. The best comparison for this is in a patient with diabetes. The reason that they have so much peripheral nerve damage and vascular damage is that their really high glucose levels over time eventually lead to glucose molecules just starting to stick naturally and spontaneously to pretty much every protein in their body. Right? They're essentially being glucosylated inadvertently. Here, C. diff produces enzymes that do it rapid speed. And so instead of taking years and years to build up that level of glucose addition, it's happening very, very fast. The place that this is happening is inside the intestinal epithelium, the cells that line the GI tract. And the specific targets for TCD A and B are enzymes in the host cell, the human cell, that control actin-cytoskeleton dynamics. And so what essentially happens is you start adding glucose molecules to these enzymes. They start losing the ability to perform their function. Actin-cytoskeleton starts falling apart inside the intestinal epithelium. And if you have had AMP or really even a basic bio class, remember that epithelial cells, this up here would be the lumen of small intestine. Down here would be blood vessels. And in order for the intestine to keep its integrity, these epithelial cells have to be linked together very tightly by cellular junctions. And when the actin cytoskeleton inside those cells falls apart, you start losing that connectivity. Cells start dissociating from each other, and what you get then is all the intestinal contents that are inside your small intestine start leaking through the intestinal epithelium and into the blood. And so you essentially get the internal blood supply of your body exposed to your intestinal contents, which is a really horrible thing. This explains the symptoms of C. diff, which is horrible intestinal damage and diarrhea. Okay, so staying in Clostridium genus, there are a lot of pathogenic species here. I'm going to compare two more, Clostridium botulinum and tetani. And it's an interesting comparison because they do sort of similar but opposite things in a way. They both affect skeletal muscle, but botulinum poisoning leads to paralysis. You can't use your muscles. We call that passive paralysis because you're just left limp and unable to contract muscle. Clostridium tetani, typo, produces tetanus. In muscle physiology, tetanus means maximum, constant muscle contraction. And so tetanus causes spastic paralysis, meaning you're paralyzed because your muscles are contracting so hard that you can't relax them and everything's locked down. And this is a good contrast in how virulence factors can have very specific effects on cells. I already went over this one in class, but I'll do it again here. The idea is botulinum toxin, right? So this is Clostridium botulinum is the species. Botulinum toxin is the virulence factor that's produced at a neuromuscular junction. Right? So this is your neuron, axon terminal. This is a muscle cell. In order for this neuron to be able to activate this muscle, these little vesicles have to be filled with neurotransmitter. And those neurotransmitters, when the muscle is activated, need to be released into the synaptic space. The idea then is receptors on the muscle should bind that neurotransmitter, and that leads to a whole cascade of changes inside the muscle cell that will lead to contraction. The details of that is an A&P topic, and I'm not going to go there. But if you can't release the neurotransmitter, it can't bind its receptor. If it can't bind its receptor, you can't activate the muscle. The way this actually works is there are these group of proteins called SNAP proteins that are required to take these vesicles, move them to the cell membrane of the axon terminal, and release neurotransmitter. These little red dots represent neurotransmitter molecules. This blue guy down here is the receptor. In skeletal muscle, the neurotransmitter is acetylcholine, and so acetylcholine receptor. Botulinum toxin, which is this protein illustrated in black right here, the thing that it does is it chops one of those SNAP proteins into pieces. Basically an enzyme that cleaves it, making it non-functional. What that means is if this SNAP complex is broken, these vesicles can't get to the cell membrane, the neurotransmitter can't be released. And so these vesicles just end up staying trapped up in the axon terminal. And so your brain can send all the signals, all the action potentials that it wants down that neuron. If you can't release the neurotransmitter, the muscle can't contract. Passive paralysis, and what you end up getting is complete loss of muscle control over time. It'll be progressive. You'll get weaker and weaker and weaker as more of those SNAP proteins are cut. The big concern is the diaphragm, because if you lose the ability to breathe, none of your other muscles really matter very much. All right, contrast to this, Clostridium tetani is producing a virulence factor, tetanospasmin, that has the exact opposite effect. Tetanospasmin is a protein that acts as an antagonist to an inhibitory neurotransmitter. The neurotransmitter happens to be GABA. It's exerting its effect in the spinal cord. And the idea here is an inhibitory neurotransmitter is normally used to keep your skeletal muscles under control. You have this constant push and pull of excitatory neurotransmitters trying to turn the muscle on, and inhibitory neurotransmitters trying to turn it off. If you completely lose the ability to inhibit the muscle, they'll just spasm out of control. And so the idea here is an antagonist is something that blocks the activity of a neurotransmitter. If you block an inhibitor, it's kind of like a double negative makes a positive, and you end up getting uncontrollable skeletal muscle activation. Okay? The excitatory signals just take over, and muscles contract so hard. Before medical treatment was available, right now, if you get a tetanus infection, antibiotics should take care of it pretty quickly. Before that was available, people would eventually die from their muscles contracting so hard they would start breaking their spinal cord and also have horrible lockjaw and not be able to eat. Okay, next one on our list is E. coli, and I've mentioned this one in class too. Most E. coli are completely harmless. It's a very normal part of almost every mammal's GI tract. It's found, pretty much ubiquitously among humans. Everybody has E. coli in their intestines. It's also a fairly common cause of urinary tract infections, but this is a good example where just picking up a single virulence factor, and sometimes a few others to go with it, can make a big difference. That virulence factor is called shigatoxin. It's just named after a Japanese scientist that discovered it first. And shigatoxin is a protein, a virulence factor, that blocks ribosomes. And so it essentially turns off ribosomal activity, inhibiting protein synthesis in target cells. The interesting thing is that serotype, the most common one is O157H1, is the thing that makes it different than non-pathogenic bacteria is simply containing the gene that codes for shigatoxin. And the interesting backstory to this is the way that strain initially got the shigatoxin gene is probably from other bacteria. There are other species of bacteria that produce shigatoxin, and it got transferred around by a virus, a bacteriophage, that actually sort of transferred or jumped the gene accidentally from one species to another. And so this is sort of another way, in addition to plasmids, that genes can potentially be transferred back and forth between bacterial species. At some point, that gene became permanently inserted into the genome of the bacterium, and so now it's stable. It doesn't have to be infected with the virus anymore. Okay. What ends up happening is E. coli can attach to the surface of intestinal epithelial cells. Same thing as the C. diff slide. This is the lumen, the interior of the small intestine. This would be blood supply underneath. And E. coli attaches to the surface of those epithelial cells. And the E. coli itself doesn't really get inside the cell, but its docking sort of coats that surface with shigatoxin and triggers endocytosis. And if you get that toxin getting inside the cell, turn off ribosome activity, if those intestinal epithelial cells can't translate RNA into new protein, eventually they're going to die. And if you get the infection cleared quickly, it's not an enormous problem. It causes a lot of GI distress and diarrhea, but you'll recover. If those epithelial cells, enough of them start dying, then the problem is the shigatoxin that was built up inside them, when these cells rupture, will start leaking out into the underlying blood supply and start affecting other distant locations inside the host. At that point, it becomes a systemic infection and inflammation in the blood, which you would call sepsis. All right, the last one I'll cover in this video, and one that also causes fairly similar symptoms, at least initial symptoms to E. coli, but through a different mechanism, is cholera. Cholera is not really prevalent in the United States or in most parts of the world anymore because we have a pretty clean drinking supply. But its mechanism is kind of interesting. So what cholera toxin actually does So cholera toxin is the virulence factor. The way that it causes such severe diarrhea, which is really how people end up dying from cholera, is only if they're so dehydrated, if they don't have access to medical treatment. It does that by interfering with the activity of a chloride channel, and what ends up happening is chloride starts spilling into the lumen of the small intestine, and if you remember your diffusion rule, water follows salts. And so if these cells are constantly leaking chloride ions into the lumen of the GI tract, water is going to follow. And if enough water keeps following, eventually patient gets dehydrated enough that they simply die from dehydration. It doesn't become some sort of systemic infection the same way that E. coli does. All right, so that's the last specific virulence factor that I'm going to cover here. The rest of these, like I said, I think it's fairly clear from the notes, and we'll go over them in class. And then when I come back with the next part of the video, we'll go on to sterilization methods and antibiotics.